Hi, hello, namaste, good evening to all. Myself Murali PG under Unacademy YouTube class. Today we are going to discuss about molecular basis of inheritance chapter. Unacademy is one of the good platform to achieve your goal in the oncoming Karnataka CET. On behalf of that, we introduce some of the courses to our beloved students, where we are focusing on Karnataka CET 2021 aspirants, and here it is with the following course packages plus course with the following features, where continuous detailed theory, MCQ, doubt clearance and mega quiz will be there. So once you subscribe, you will going to get unlimited access to all the educators topic. During the subscription, use my referral code or use code, it is KCET Murali, and get 10% off with respect to the price values. That is one thing. And secondly, the comprehensive batch. Myself, I am taking this comprehensive batch, and here it is complete syllabus is going to be covered. Tips and tricks by top plus educators. Every topics will going to have practice questions. Full length mock test also be there. Use my code KCET Murali and get 10% off on this class also. Mestros batch, it was introduced on 15th of December. I am taking this Mestros batch from 7 to 8 p.m. every day, and here also dedicated doubt clear session, MCQ, theory and mega quiz etc. will be there to the respected students. And comprehensive batch, another module, and best plan for Karnataka CET, where 12-month plan and 6-month plan. If you are going to compare the price, this is better, and 6-month full syllabus, dedicated doubt clear session and completion in Kannada language also if you want. Then multiple batches launching every month, so that will going to fulfill the requirement of our students. Next, best plan for KCET 2021 aspirants, multiple batches launching every month. Okay, and daily T20 free practice test for Karnataka CET. So it is for free and started on November 2nd. And here you can use it, and as a result of that you can get the benefit from our side. So your preparation will be in better way to face the oncoming Karnataka CET. So that is what my important notice towards student. And also I am taking special classes. You do one thing, download Unacademy Learners app from the Play Store, install it, and later search under goal for Karnataka CET KCET. Under that, give my name Murali PG or use my code, that is KCET Murali, and you can have the access to my special classes. And also for subscription, you have to use my code KCET Murali and get 10% off with respect to subscription price. Okay, but a special class, it is for free. You can get that one by installing the Unacademy Learners app. Go to goal, under that Karnataka CET KCET, and select my name Murali PG or use my code KCET Murali. Okay, you can access to the classes of special class category. Okay, so by this brief introduction, now we are entering towards the important MCQs discussion under our molecular basis of inheritance. Under this I added about 30 questions. Okay, so welcome to all my dear students. Now let us have the discussion of questions. Okay. Manoj, welcome to the class. Good evening. Manohar, welcome to the class. Good evening. So, first question of this molecular basis of inheritance here is which additional group is present at the second position of the ribose sugar in RNA. So why it is called deoxyribonucleic acid? DNA is not having that additional group in second position of ribose sugar. Second position with respect to second position carbon atom with respect to pentose sugar of RNA. So DNA it is not there. In the DNA it is not observed. In the RNA we can observe that one. So which is that one? That is OH. Additional OH group that will going to give that deoxyribonucleic acid name or that nature. Okay. First question answer is C. Second question. Thymine is also called what? Thymine. So we are having adenine, guanine, cytosine, thymine, isn't it? Sorry. Now thymine is also called what? Alternate term for thymine. That is what? 2-methyl uracil, 4-methyl uracil, 3-methyl uracil, 5-methyl uracil. The right answer is 5-methyl uracil is the right answer. Okay, thymine is also called 5-methyl uracil chemically. Then in DNA molecule, if cytosine is 18%, then the percentage of adenine would be how much? Cytosine. Yes, Rakesh, welcome to the class. Good evening. You are right, Rakesh. For the previous question, it is D is the right answer. 5-methyl uracil. Yes, for the third question, in the DNA molecule, if cytosine is 18%, then the percentage of adenine would be how much? How much? 18, 32, 36, 64. Yes, Devesh. Yes, for second, D is the right answer. What about third? What about third? Cytosine, if it is 18%, what about the percentage of adenine? Cytosine will going to form the bond with thymine. Guanine with the, adenine with the thymine, guanine with the cytosine, isn't it? So cytosine bond with the guanine, GC bond, where 3 hydrogen bond will be there, and adenine pair with the thymine by 2 hydrogen bond. So now the amount of cytosine is 18%, and what about the percentage of adenine? So which is the right answer? Yes, any answer? Then the percentage of adenine is how much? (32, 64, 36, 18). How much? It is B, 32. How it will become 32? Here, based on Chargaff base equivalence rule, number of purine refers to, here it is adenine and guanine is equal to number of pyrimidines, that is cytosine and thymine. Hence the amount of guanine should be equal to the cytosine. Adenine should be equal to thymine. As per the data given above, here the percentage of guanine may be 18% because cytosine is 18%. Total percentage of adenine and thymine is equal to 100 minus 18, that is 64. So the percentage of adenine is equal to 64 by 2, it is 32. That is what the answer B is the right one, 32. Okay. So answer for that question number third, it is B. Okay. Next, fourth question. In A, B, C, D and Z forms of DNA, which one is left-handed? It's not left, it is left-handed. Which is the left-handed DNA? Right-hand helix and left-hand helix. There are two types. So in A, B, C, D and Z forms, different forms of DNA. In that, out of 5 types, which is the left-handed DNA? So it is Z form of DNA is left-hand helix. So right-hand helix and left-hand helix, according to the helical coiling with each other, it is left-hand and right-hand helix formation. So the Z form of DNA is showing left-handed helix. Next, question number 5. Find out the number of base pairs in E. coli, that is E. coli DNA, if its DNA is 1.36 mm long length. Number of base pairs is how much? It is 4 into 10 to the power 6 base pair. So by converting its mm into micrometer, you can get the answer. So by using the statistical approach, the right answer is A for fifth one. Then sixth one, arginine and lysine are A proteins and are found abundantly in B. So arginine and lysine are considered as one particular protein and found in maximum percentage in B. So which is that? What is the option for A and B? Yes, for 5 it is fifth. Manoj, you are right. Devesh, welcome. Yes, for fifth one A is the answer. For sixth one, what about the answer? It is B. Okay. So arginine and lysine are basic protein with amino acid and are found abundantly in histone protein. Histone protein is made up of amino acid arginine and lysine. Histone proteins are positively charged particle. Around its negatively charged particle, DNA is wrapped. So histone octamer, around this histone octamer, we can observe negatively charged DNA particle is wrapped. So that is what our nucleosome model, isn't it? So histone proteins are with the arginine and lysine, they are basic, whereas DNA is acidic in nature. So protein, that is histone protein, it is basic and positively charged, whereas DNA molecule is a DNA which is negatively charged particle. Okay, that is what the answer for sixth one. B is the right one. Okay. Next, seventh one, lightly stained part of chromatin which remains loosely packed is which one? So according to the chromosomes, chromatin were lightly stained part and darkly stained part, or lightly stained chromatin and darkly stained chromatin. Here for that, if you take the concept of our particular chromosome theory or chromatin, there the terminology is euchromatin, heterochromatin, Chromatosome, chromonematome. It is the euchromatin. Euchromatin is a loosely packed and lightly stained chromatin. So right answer is A for 7th one. Then 8th question. 8th question. Yes, 7th one A. Lahari, you are right. Welcome to the class. Good evening. Devesh, welcome to the class. Good evening. If you have any doubt, you can ask. Okay. Yeah. Now the answer. In Griffith experiment, what would be the effect of following conditions on mice? So Griffith experiment. From form of pneumonia injected and effect on mice. Live, rough, non-capsulated A. Live, smooth, capsulated B. Heat killed smooth C. Heat killed smooth plus live, rough D. Representation of these injections. Choose the correct option for the effect on mice. Which are the options? Correct option. Which is the correct option? So 8th one, according to the requirement. Here right answer is B, means A is survived. Live, rough, non-capsulated. It is not going to show any virulence. It is not going to show any kind of disease with respect to mice. Pneumonia is not there, so it will survive. And second one, live, smooth, capsulated. Live smooth is virulent, going to cause pneumonia, and mice is going to be died. So death will be there. And third one, heat killed smooth. So when the heat killed smooth variety is there, the cell is not survived, but genetic material may be there, but it is not going to be showing any virulent nature. So survived. And last one, heat killed smooth plus live rough, where the mice will going to die because of transforming principle. Heat killed smooth, genetic material is still intact. It will going to convert live rough strain into smooth strain, result in the formation of or occurrence of that pneumonia fever in the mice. So mice will going to die. So right answer is B for 8th one. Next, question number 9. Who gave an unequivocal proof that DNA is the genetic material? By whom? So DNA is DNA is genetic material. The proof was given by whom? Griffith, Avery, MacLeod and McCarthy, Alfred Hershey and Martha Chase, Mendel and Watson. So right answer is C. Alfred Hershey and Martha Chase, and commonly the well famous experiment, it is called what is that experiment? Hershey and Chase experiment, where DNA is the genetic material. Proof was given by the scientist, okay, a group of scientists, that is Hershey and Chase. So experiment is called Hershey and Chase experiment. Next, question number 10. What was unique in Griffith experiment? Unique feature of Griffith experiment. DNA was found to be genetic material. RNA was found to be genetic material. No. Something from dead organism could change the living cell. And the fourth one, viruses can live in bacteria. So right one is C. Something from dead organism, that is heat killed S strain will going to convert R strain into S strain. That is what? Something from dead organism could change the living cell. Which is this living cell? That is R cell. R cell is converted into S cell. R strain cell is converted into S strain cell. So answer for the 10th one, it is C. For 9th one also it is C. Hari, you are right. So that is a transforming principle of Griffith. Next, 11th question. Which group present in RNA nucleotide is very reactive and makes RNA liable and easily degradable than DNA? Why RNA is not stable like that of DNA? What is the reason? The reason behind this one is 2 OH group on ribose sugar. So additional OH group in the ribose sugar complex, that will going to give the nature to the RNA nucleotide with very reactive and more liable and easily degradable when comparing with the DNA molecule. So right answer for 11th question is B. Then DNA and RNA both have which kind of molecules? Adenine, cytosine, phosphate, guanine and uracil. Choose the correct option. DNA and RNA both which are the common molecules. So it is D is the right one. The first one adenine, sorry, not D, it is one second, adenine, one second we will see, guanine, yeah, guanine, then third one cytosine and phosphate, not uracil. Okay, so it should be 5. Okay, here 5, 1, 2, 3rd and 5. Okay, so right answer for 12th one, it is D. So adenine is there in DNA and RNA, cytosine is there, phosphate is there, guanine is there, uracil is present in the RNA, thymine is present in the DNA, isn't it? So right answer is D for question number 12. Yes, Manoj, for 11th question you are right, B is the right answer. Next question, number 13. Which of the following molecule is most susceptible for UV radiation? Which is that one? RNA, DNA, both A and B, protein. Which is more susceptible? That is RNA. That is liable, more reactive, more unstable. That is the RNA when comparing with the DNA. So right answer is A. 14th question. DNA dependent DNA polymerase catalyzes polymerization in which direction? Polymerization means what? Production of another DNA molecule. In which direction? 3' to 5' here, 5' to 3', 5' to 2' and 2' to 5'. Arrow mark should be there. So right answer is C, 5' to 3'. Next question number 15. Here the question is deoxyribonucleotide triphosphate serve dual purpose. Which are the dual purpose? Deoxyribonucleotide triphosphate is going to have dual purpose. The purposes are 15th one, that is answer is D, refers to acting as substrate and providing energy for polymerization reaction. Okay, acting like substrate for the production of new DNA strand and also providing energy for that polymerization of DNA. So right answer is D. First and third statements are right. Next question number 16. Okay, so Manoj, thank you. So heavy DNA can be differentiated from normal DNA by which centrifugation technique? Heavy DNA molecular weight is high with the normal DNA. How we can differentiate? By which method? It is called cesium chloride density gradient method. Cesium chloride density gradient centrifugation, you can say like that. Cesium chloride density gradient centrifugation is a methodology to separate out DNA, heavy DNA and light DNA or normal DNA. So question number 16, C is the right answer. And 17, match the following columns. In the first column I given different enzymes, and in the second column the function of that enzyme. So here I will give directly the answer. D is the answer, refers to topoisomerase. It is relaxes the DNA from its supercoiled nature. DNA gyrase relieves the strains of unwinding by DNA helicase. This is a specific type of topoisomerase. DNA ligase reanneals the DNA ligase it is for joining of Ogasaki fragments of lagging strand, that is a DNA ligase. Reanneals the semi-conservative strands and joins the Ogasaki fragment, that is right. And fourth one, D is the primase, provides a starting point of RNA for DNA polymerase to begin synthesis of the new DNA strand. So during that time it requires the starting point, it is by using RNA small strain. Then telomerase is the lengthens telomeric DNA by adding repetitive nucleotide sequences to the end of eukaryotic chromosomes. So right answer is D for 17th one. Yes, Rakesh, you are right. Okay. Next question number is 18. Free a OH of which prime? Okay, A means which prime, whether 4 or 3 or 5 or 2 of RNA, OH of RNA primer provide the initiation point of DNA polymerase for sequential addition of deoxyribonucleotide. Identify A, which is the prime, in which prime it will going to be start. It is the C3 prime OH. Okay. Minus sign and plus sign of for the DNA strand stands for what? Minus and plus strain. It is for 19th one, D. Non-coding strand and coding strand, template strand and non-template strand, anti-sense strand and sense strand. All the three statements are right. So right answer is D. Then monocystronic transcriptional unit codes for what? Monocystronic transcriptional unit codes for what? Mono, single, cistron, cystronic transcription unit, that is coding for single polypeptide chain. Then 21st question. A gene that takes part in synthesis of polypeptide. Which is that gene? It is structural gene. Of course, structural gene code for a polypeptide, not promoter or regulator operator. So if you take a lacoperon, isn't it? 22nd, hnRNA, heterogeneous nuclear RNA, undergoes additional processing called what? Before the complete maturation of mRNA in the case of eukaryotic transcription, before the beginning of translation, mRNA is going to be synthesized and it is undergoing some processing. That is called what? Capping, tailing, both A and B, none of this. 22nd, it is C, both A and B. We can observe capping and tailing. So addition of poly A tail and capping phenomena will be there. So as a result of that, heterogeneous nuclear RNA, after the release of that or after the detachment of that intron from exon, so exons are joined and capping and tailing will be there. After that only that eukaryotic mRNA will enter into cytoplasm from the nucleus for translation purpose. It is observed, this kind of phenomena is observed only in the case of eukaryotic. In the prokaryotic, the spliceosome phenomenon is not this. Spliceosome refers to removing of intron, joining of exon and capping and tailing. Okay, total process. Then methyl guanosine triphosphate is what? Why it is going to be utilized? It is nucleotide used in capping. Just in the previous question I said capping and tailing. In the tailing we can observe polyadenylation. Adenine group is going to be added, many hundreds. In the cappings end, methyl guanosine triphosphate is added. So these two, capping and tailing, will provide total protection to that entire stretch of mRNA when it enters to the cytoplasm. Then starting codon is in the prokaryotes codes for what? So here should be in prokaryotes codes for what? So starting codon with respect to protein synthesis, it is formyl methionine. tRNA F-met, isn't it? tRNA F-met is the starting codon that codes for particular methionine group, which will carry that methionine group. Okay. Which of the following pair is correctly matched? So correctly matched pair is D. Anticodon, site of tRNA molecule that contains complementary bases to the triplet codon on mRNA. So mRNA. Okay. So anticodon, site of tRNA molecule that contains complementary bases to the triple code on the mRNA. So right answer is D. Next, positively regulatory proteins are called what? Positively regulatory proteins are called what? It is the activator. This is called activator. And 27th question, RNA primers are removed by which enzyme? RNA primers during the DNA polymerase reaction. So DNA polymerase chain formation, DNA formation, RNA primer is going to be removed. It is done by which enzyme? That is a DNA polymerase enzyme. Okay. Then percentage of similarities between the nucleotides of two individuals is how much? 28. It is C. 99.9% is alike, same with respect to human being, isn't it? So right answer for this question number 28, it is C. And 29th, last two questions. For the given DNA sequence, 3 prime TACA TGG GTC CG S. Sorry, it's not S. Okay, it's not required. 5 prime. So 3 prime to 5 prime. Choose the correct code of tRNA anticodon. Which are the tRNA anticodon? So here we have to write, isn't it? So for 29, D is the right answer. Means first AUG barate. Here AUG. Then UAC. CC and A. Then last one GGC, isn't it? So right answer is D for question number 29. And last question, shape of 3D view of tRNA is what? 3D view of tRNA, it is what? So right answer is 3D view, it is L shape. Okay. So answer only fourth one is the right one for the 30th question. So by this I am going to end up this session. Okay. So if you want more, use this plus subscription by using my referral code or use code, that is KCET Murali, and use this code to get 10% off with respect to the price value. And there is a comprehensive batch, Mastros batch, and the champion batch is also the best plan for Karnataka CET 2021 aspirants. Okay. And also daily T20 free practice test will be there. You can use it. Okay. So thank you. See you again in the next class. Thank you one and all.